we realize that enzymes, what they do is they break down large molecules into smaller molecules. Now, we learned about them first when we learned about digestion. That's the first time, but they're not the only enzymes that exist. And later on in your GCSE, you're going to learn about other forms of it. But to start off with, let's think about it just in terms of digestion. There are lots of different types of enzymes, and each enzyme is designed to break down a particular different molecule. So, for example, amylase is one type of enzyme, and it is designed to break down starch. When it breaks it down into, well, at GCSE you learn that it breaks it down into glucose. But what it looks like here is it actually first breaks it down into maltose, then something else breaks it down into glucose. But in any case, starch, amylase is used to break down starch. Okay, so glucose is a much smaller molecule than starch. So why do we want to break them down in the first place? Why do we want to break down starch into glucose? So the reason why starch is not as useful as glucose essentially is because starch is a larger molecule. Now, the problem with it being a larger molecule is that we can't absorb it into our blood as easily. Why do we want to absorb it into the blood? Because the blood's going to transport it around the body and eventually allow it to get into our cells. So we want to get the glucose into our cells. We can't get the starch into our cells because it's too large. So we need to first break it down. And the thing that helps us to break down is the amylase. Okay. So yeah, different enzymes break down different molecules. So amylase is good at breaking down starch, and the reason for breaking down starch is because starch is not so good at getting into the blood, and we need to get it into the blood in order to use the molecules. The molecule in this case is used to gain energy. So if we chop it up using the enzyme amylase, we first chop it up into maltose, and then it looks like another enzyme will come along and chop it up again, and that chops it up into a glucose molecule. The glucose can get into the blood, and so now we can use it. So the whole point is to chop it into smaller molecules, which we can then use. So I said that other enzymes, let's go over here. Other enzymes actually break down other molecules. So we said, so amylase breaks down starch into glucose, which what does it do? It helps to digest food. Okay, well, yeah. What it does is we want to just say it breaks down. The sentence just wants to say we break down starch into glucose, and
Yes, I forgot.
there are definitely more than three, but these are the three main ones to do with digestion. There will be other ones as well.
Ones that I'm
I can't guarantee that these are the only ones you'll need for your test, but I think the main thing is to understand what enzymes are and how they work and so on, and then we can start to learn other examples of them later on. So you can also have protease, which breaks down protein into amino acids, and then we also want to know where these enzymes can be found. So stomach, pancreas, and small intestine. So that the other thing we do want to know about enzymes, though, rather than apart from what other types of enzymes are there, is about something called denaturing. So I wonder if they have some notes in here to explain it. Let's have a look at this. This is okay. Let me just put it on pause and I'll find something. Right, so yeah, going back to your question about what other enzymes are there? Well, you can see that we have different enzymes. For example, shrimps have a certain enzyme to do whatever they need to do. But the point is, these enzymes are designed to work in particular situations. So you're going to find that our enzymes are very well designed to work at body temperature because that's the temperature that they work in. So when they so I think you'll probably find the red one is probably going to be I don't know what these enzymes are, but it's probably going to be an enzyme that exists in humans. So you can see that around about body temperature for humans, which is 37 degrees, we have the enzyme working really well. The rate of reaction is really high, and then outside of that temperature, it drops off. So that's the same with pH. They'll have a preferred temperature, a preferred pH, and a preferred something else. I forgot what the ones are, but temperature and pH are the main ones. Let's just find let's do a little bit of, find some information about preferred other things that they need. So looking on BBC Bite Size, I think this bit talks about the different factors, and this is what I was showing you. We have this optimum temperature, the temperature at which the enzyme would prefer to work at. Now, first of all reactions react more quickly. All things react more quickly when they're hotter. However, with enzymes, they're going to prefer a particular temperature because beyond that temperature, they denature. I was talking about denaturing. Hopefully we can have, here we go. So this is how they would normally work. We have this active site. The substrate is a thing that we're trying to break apart. And as you can see, a nice healthy enzyme working at the right temperature, the right pH and everything is a nice fit for that particle, for that molecule. So it'll slot in place and then we can make it react. But when we increase the temperature, what happens is we say that the enzyme is denatured. And what this means is it changes shape so as no longer will that fit inside and they don't connect properly, and so the reaction doesn't happen anymore. So you have this situation. Beyond a particular point, the activity drops off because it doesn't. The enzyme has been denatured, and it doesn't react anymore properly. Does that sort of make sense? So why don't you let me just save that video there.